Bonjour à tous. Donc, nous allons pouvoir démarrer cette édition des webinaires des MétaSP en herbe. Donc, avant d'écouter la conférence du jour, je souhaitais prendre 5 minutes au nom de toute l'équipe déjà pour vous remercier d'être là aujourd'hui pour la rentrée des webinaires des MétaSP en herbe. Donc, je vais également rapidement vous introduire la nouvelle équipe des MétaSP en herbe qui va œuvrer cette année pour la préparation et l'animation des webinaires. Donc, à ce jour, nous sommes 5 doctorants de différents labos, chacun travaillant sur des thématiques plus ou moins différentes et complémentaires. Et nous vous avons déjà préparé un programme de webinaire que vous voyez à l'écran, que nous vous avons fait suivre afin d'attiser un petit peu votre curiosité et vous inciter à nous rejoindre pour les prochaines éditions qui aborderont des thématiques variées, toujours autour du métabolisme spécialisé. Donc, bien sûr, je terminerai cette petite intervention par la conclusion habituelle. Donc, si vous êtes intéressé pour présenter vos travaux, ou bien si vous êtes jeune chercheur souhaitant intégrer cette équipe, vous pouvez nous contacter à l'adresse de votre choix. Quant à moi, je vous souhaite à tous un bon webinaire et je laisse la parole à Léa.
Donc, bonjour à tous. Hello, everyone. Thank you all for being here for this new edition of MétaSP en herbe, which will focus today on the plasticity of the seed specialized metabolome in wild and cultivated camelina species. And so today we are pleased to welcome Massimiliano Corso for this conference. So Massimiliano completed a thesis in the University of Padova, so in Italy, during which he studied the role of specialized metabolites on grapevine tolerance to abiotic stresses. He then carried out a postdoc at the Université Libre de Bruxelles in the team of Nathalie Verbruggen, where he worked on the role of flavonoids in metal homeostasis, and then he continued on the team very close to this one by integrating the team of Sébastien Tomini in the ITBC as a postdoc too. And so then he was recruited in 2019 as a researcher at the Institut Jean-Pierre Bourgin, so in INRAE Versailles Center. And so now he's working on the diversity and induction of the seed specialized metabolome, a subject that he's going to present right now.
So thanks, Léa. Merci, Léa. So I will do the presentation in English, as in theory should be only some English non-French speaker in the audience. Then, of course, if you want to ask some question in French, it will be okay. So thanks again for the introduction. So today I will talk you about a part of the work that I am developing in the team, in the CDEV team of the Institut Jean-Pierre Bourgin, the INRAE Center in Versailles. And more specifically today, I will focus the attention on the diversity and the plasticity of the seed specialized metabolome in wild and domesticated camelina species. So very quick introduction on the specialized metabolome. I think you don't probably need it, but just in case. So as you well know, plant metabolites are divided into two main macro categories. We have on the one hand what we call the primary metabolites, which are, for example, amino acid and protein, lipids and sugars, for example. Some lipids, or what we call usual fatty acid, are also included in this category. And then we have the specialized or secondary metabolites. Here we have some example as the flavonoids that we know very well in our team, glucosinolates, and again other lipids, or what we call unusual fatty acids. So what does it mean? In general, we say that the primary metabolites are inward oriented, so they are essential for the viability of the cell, the growth, the development, and the reproduction. And we say that they are indispensable. It means that they are indispensable for all the processes that I just cited. There are hundreds of metabolites that belong to this category, and they are generally highly conserved among plants, at least for what we call for a part of them. In other case, we have other kind of metabolites that are named specialized metabolites, as you know, that they are outward oriented. It means that they are necessary for the viability of the organism within its environment, and we say that they are dispensable. So we know that there are some metabolic pathways that are not activated in a certain condition. Let's call them control condition or normal growth condition, and then very rapidly activated when there is a perturbation, a stress, or something that changes the environmental level. There are thousands and thousands of metabolites that belong to this category. They are much higher number compared to the primary metabolites, even if, of course, the primary are the precursor of many of these specialized metabolites. And what is important is that they are highly variable within species and also genotype of the same species. So again, as you well know, there are three main major classes of specialized metabolites. We have on one hand what we call the triterpenoids, with around more than 30,000 compounds, and the number I'm changing so fastly, so probably there are much more than that. Then we have a second category with the alkaloids and the nitrogen-containing molecules. That's, for example, the glucosinolates that are specific to Brassicaceae species, such as Camelina sativa that I will present to you today, and other kind of alkaloids. And then there is a third category. That are the phenolic compounds or the phenylpropanoids, that our team is studying till a very long time, way before my arrival in the team. And today I will focus a part of the presentation on this category. So we are in the team, we are studying and also my research is mainly focused on the diversity and plasticity of specialized metabolites in the seeds. So why the seeds? Let's start from the fact that seeds has a very different specialized metabolome compared to other organ of the plants. Here you have two PCA that are showing the distribution of different organs and tissues that according to their specialized metabolic diversity profile, and as you can see, either in Arabidopsis, a non-cultivated and model wild species, and the same is true for cultivated species such as rice, the seed has a very different specialized metabolome compared to what we have in the other organs. So why seed specialized metabolites? Do they have a role? Why they are important? So in general, this is true for seeds, but also for other specialized metabolites in other part of the plants. We don't know that much about the role of seed specialized metabolites, so that's why I think it's really interesting to study their diversity and plasticity. What do we know is they are somehow involved on the one hand in seed responses to abiotic and biotic stresses. So abiotic stresses such as high temperature or drought, or biotic stresses such as pathogen resistance. And of course, then they have also a very important and key role in nutrition and in general in human health. So many of these specialized metabolites such as flavonoids, but that's not the only category. They play a role in nutrition as anti-malarial, anti-cancer, or more generally, we say that they are antioxidants. So they can counteract, somehow respond to the accumulation of reactive oxygen species. So Okay, so while so this is a very quick slide to tell you why we study this metabolite in seeds and why we are interested in the elicitation on the induction of the specialized metabolome in seeds by stress. We know that most studies focus on specialized metabolites in plants. Most studies focus on specialized metabolites in vegetative tissues, while the studies of seeds, of the ability of seeds to produce these metabolites, were somehow neglected or not well studied. So my research in our team is really focusing on this subject, so on the diversity of seed specialized metabolites and on the regulation, on the impact on the environment, let's say, on their accumulation, regulation, plasticity, and etc. So today I will focus on one Brassicacea species that we love at IGPB, which is camelina species. So as I told you, is a Brassicacea such as rapeseed, for example, or the model species Arabidopsis. Why is study? Why is sort of a kind of relevance in the or attract attention in the last in a few past years? Because it has a unique oil composition. In particular, it is characterized by high level of polyunsaturated fatty acids such as omega-3. It is characterized by adequate yields under low input management and in limiting environments, so it doesn't need a lot of water, for example, for their cycle, and also the other inputs are quite limited. So as an example, we cultivate some camelina genotypes this summer. It was quite a hot summer even in the Paris region, and the plants, I mean, behave well with very, very limited or no amount of external water. So it's very cool. It's a plant that we can transform. It's a very nice model. It has a short cycle compared to rapeseed. It doesn't need vernalization, at least some accession, but it is an hexaploid. So what does it mean? It has a very complicated genome compared to other species. And here you have an example and a citation of a very, very nice paper that showed how at least to show how the evolution of the camelina genome. So in our case, what we are using in these studies are two species of camelina, Camelina sativa and Camelina microcarpa. Sativa is the domesticated species, so the one which is used, let's say, in agriculture and for oil production, and microcarpa is the wild species, so the non-domesticated one. And as you can see here, there are several parents of these two species, or this is what we think that they are the parents of these two species. And we think that the sativa and microcarpa species came from two parents that are Camelina neglecta, which is a tetraploid, and Camelina ispida, which is a diploid. So basically, to make the long story short, when you have one gene copy of, for example, in Arabidopsis thaliana, you have always three genes in Camelina sativa. So as you can imagine, if you want to follow a genetic approach to study the function of a gene, is a bit more complicated. So we have to knock out three genes at the same time, free copy. But still, it's a very convenient model and interesting to study. So today I will present you two main studies of that are ongoing, and one is already finished in the lab. So the first one was made by Belea Barreda, which is a PhD with me, that she is also one of the organizers of this very nice cycle of seminar, and Stephanie Boutet, an engineer which is working in the metabolomic platform of the IGPB. So the first work is about the untargeted metabolomic analysis in different Camelina sativa genotypes. So what we did, this is a collaboration we have with some friends from the University of Bologna, in particular Federica Zanetti, that provide all the plant material And what they did, our colleague in Bologna, they grow 6 different varieties or genotypes of camelina sativa that came from different breeding programs. They cultivate these varieties in the field for 5 consecutive years, from 2015 to 2019. Then what we did after, the dry seeds, so the mature seeds at the end of the cycle, were collected and were initially subjected to LC and SMS analysis to study the untargeted, the polar and semi-polar metabolites. And we perform also untargeted lipidomic analysis. In addition, we also perform, in collaboration with our colleague from the IPS2 Institute, the analysis of some primary metabolites with GCMS. So the objective of this study was to study the environmental induction and the genotypic signature of C-specialized metabolites in camelina sativa species. So what do we know about the meteorological condition already where this plant grows? So as you can imagine, as I said before, there are several studies that focus on the vegetative part, and in particular, what is the role of the environment, of these environmental stresses in the induction of the pathway, the metabolic pathway in the vegetative tissues. So here also we wanted to have a look at the impact of the environment on seeds metabolites, and in particular also why is in seeds that are very important in many brassicacea and oilseed species, where they constitute the final product basically that are used for the industry, for agriculture, for food production, oil production, etc. So knowing and studying the quality of these seeds and the impact on the environment on their quality is very important. So here the plants were grown in the same site close to Bologna, so in the center of Italy. So as you can see here with the 3 colors, you have the low, the average, and the higher temperature. So there were the average temperature from 50% flowering to harvest. So as you can see, there are quite some differences if we compare the different years in terms of the temperature. And also what you can see also here is that this bar is indicating the precipitation. So there was a year 2016 where it was characterized by very high precipitation, and others such as 2017 where the precipitation were much lower. So first step was to study the impact of the environment, of the years, or of the genotype on the oil content. So we measure the total oil content in all these samples, so the 6 genotypes over the 5 years with 4 biological replicates, and then we didn't observe a very strong impact on the environment or the genotype on the content, on the oil content. The same was true for protein content, even if there were there was some kind of induction, of protein accumulation in 2017. That was this year with lower precipitation, but still the impact was quite moderate. Then what we did, we performed untargeted metabolomic analysis with a cutoff. So with no big surprise, many metabolites were not annotated, where it doesn't has a clear annotation according to homemade or public database, which is something which is well known, let's say. But still, we were able to annotate, or better, to assign to a metabolic class as a part of our metabolites. So how do? We use a strategy which is called a molecular network. So you will have a very nice presentation in one of the next seminar by David Tubul that he will explain, I'm sure, much more in detail about how does it work, this technique. But basically, what do we have here in this network? We have each node, so each circle is a metabolite or a metabolic features. And when the metabolic feature has a similar structure according to their MSM spectrum, they are clusterized together. So here, for example, we have a cluster that of flavonoids. So it means that many or some of these nodes are annotated as flavonoids because we have standards or because we have a match with a public database. So we were able actually to assign the same category to all the other metabolites. So I really simplify the procedure, but this is how does it work. So this technique is very cool because it really allows to improve the quantity and quality of the assignment of a metabolic class of all the features, and also the annotation with other software that we use in the lab. So among the metabolic classes that were identified in the camelina dataset, many were flavonols, and other were amino acids and derivatives, cinnamic acids, carbohydrates, etc., etc. There were few glucosinolates number compared to what we observe in other species, such as Arabidopsis thaliana, or also in rapeseed species, for example. But even if the number is low in terms of diversity, let's say, the quantity is relatively high compared to other species. So there are few glucosinolates that are highly accumulated, basically. And so what do we do with all this information? That's cool. We have our data set. We have the annotation of the assignment of to a metabolic class of many features. Next step is to study, to evaluate the impact of either the environment or the genotype on the accumulation and diversity of specialized metabolites. So to make a long story short, we did sort of an ANOVA test with a pairwise comparison at each year and variety. And what we observe is that, okay, at least there is a huge impact either of the genotype and the environment on seed specialized metabolite diversity, but the impact on the environment, the environmental conditions, so the impact of the different years, let's say, of growing, which are characterized by different temperature, precipitation, etc., was very strong. So actually 93% of the total specialized metabolites detected in specialized metabolic features detected in our dataset were somehow affected by the environment. So at least they were more or less accumulated in at least one year compared to the other. So that's cool. So the next step was for us to trying to identify which metabolic classes were present in this data set. So we identified 3 annotated metabolic classes that were presented here, namely the flavonols, the glucosinolates, and the amino acids and derivatives. Among all these classes, I won't go too much in detail, but we perform a correlation analysis among all the metabolites to observe whether there were different behavior, so accumulation pattern, let's say, in different years or in different genotypes. And what it popped out is that among all the annotated metabolic classes, we identified one, which are the flavonols that were highly plastic, let's say. So they show many different accumulation patterns over the different year and the genotype. So the next question was, we know that the flavonols, they are definitely some phenylpropanoids. They are important in plant interaction with the environment, at least in the vegetative tissues. What we asked to ourselves is that whether the impact, whether there was an impact of the environment on the accumulation of these metabolites and more specifically on their decoration. So the question is, does similar environmental condition also induce the accumulation of metabolites with similar structure? So that's the question. So we try to answer to this question, and we identified actually some metabolites, some metabolic and some decoration of these metabolites that were indeed showing a clear signature according to the environment. Here you have one example. For example, in 2016, that was characterized by higher precipitations, among, of course, all the other environmental conditions. We observe a higher accumulation. So here you have a box plot representing the accumulation of many specialized metabolites that are showing that are derived from quercetin, flavonoids, and are showing this kind of decoration, glucoside with a malonyl or acetyl phosphate group. And also we observe many quercetin derivatives that show triglycosylation of their structure. Here another example. So we have 2019 there was a year characterized by higher maximum temperature compared to the others. So here in 2019 we observed many metabolites that were on one side related to their derivatives of camphorol flavonoids, and there were glycosylated and malonylated. So it presents true decoration in position 3 here. And another example of another year that was also characterized by higher temperature compared to the other, but lower than 2019. We observed many metabolites related to quercetin, in particular with triglycoside plus coumaroyl or feruloyl group. So that's just an example, of course, of what we observe. But in general, so we observe in many cases that the environmental condition was sort of driving the accumulation of some metabolites. So this is basically the take-home messages. Many metabolites, and more important, many decorations, so glycosylation, acylation, etc., seems to be somehow driven by the environment. And in particular, here you have a classical biosynthetic pathway of flavonoids. In particular, here I focus the attention on flavonoids that show a very huge diversity, but this diversity basically is really driven by their decoration. So we have relatively few aglycone that we can identify in camelina species. They are mainly quercetin and camphorol, but we observe a huge diversity of these metabolites, which is mainly driven by their glycosylation, methylation, and acylation and hydroxylation. I forget to mention here patterns. So somehow this decoration, of course, as you know, they are influencing the diversity of the metabolites and also their, of course, bioactivity, transport, etc. So we arrive at this step. We show that many metabolites were specialized metabolites were affected by their decoration patterns and were affected by the environment. We then perform other analyses to study more precisely the untargeted lipidomics and the analysis of fatty acids and also primary metabolites to compare what we call the plasticity, or the ability to accumulate some metabolites in response to the environment. So what we observe, so here these are the box plots that are showing the plasticity of each metabolic classes. When the plasticity is high, it means that it's very responsive to the environment. The index is close to 1. When the plasticity is very low, it means that it doesn't respond a lot to the environment. The metabolic category does not respond a lot to the environment. The plasticity is very low. So here we identify among the specialized metabolites many metabolic classes that were characterized by moderate to high plasticity. This was true, and this was partially a surprise for some free amino acids and carboxylic acids, but to a much lesser extent to sugar. While many primary metabolites, there are many reserve compounds that are present in camelina oil, such as many fatty acids, the most important one, and most of the lipid classes, such as diacylglycerol or triacylglycerol, and also protein content were characterized, but much lower plasticity. It means that they do not respond a lot to the environment, which is kind of a good news in this case, because okay, we use camelina for its oil production, for its oil quality. So the fact that many of these compounds are stable among different years and according to the environmental condition is, in my opinion, is a good news. So to summarize some part of this work, I show you that there is a strong effect on the environment on seed specialized metabolites. So not only the vegetative tissues are responsive to the environment, but also seeds show a very plastic and dynamic specialized metabolome. Flavonol decoration seems to be somehow influenced by the environment, but this is true also for other category of specialized metabolites. And we show that specialized polar and semipolar metabolites show much higher plasticity compared to other reserve compounds, such as many lipid and fatty acid classes. So then very obvious questions arise from all this work. So if seeds are able to shape their specialized metabolome in response to abiotic and biotic stresses, and if yes, what is the impact of these stresses on the accumulation of the metabolites and the role of the metabolites in response to stress, if there is an activation by stress of the metabolic pathways. And this is just to do the transition for the second war. I will be fast. I will try to be fast. Where we are investigating the genetic diversity and induction of the specialized metabolites in camelina species. So the work is carried out by Stephanie and Leah, and also by Nicola Klevko, which is an engineer in our team. And this work is financed by a Plan to Pro project, which is named Brassimet, in collaboration with some colleagues from Rennes. So very quickly, what was the rationale behind this work? So we want to induce somehow some responses in, try to see if we were able to induce some metabolic responses in camelina seeds. So actually want to induce the metabolome in camelina by somehow mimicking some stress. So we choose a copper chloride treatment, which is also used to prime some defense response in plants against biotic stress. We use it at a relatively high concentration also to induce a sort of stress in the plants and in the seeds. So here are the effect of the treatment of copper chloride. So we start to use copper chloride treatment to spray it at the onset of flowering in camelina plants and seeds and calyx, and we perform one treatment per week. We use two different species. We use the domesticated species Camelina sativa and the non-domesticated species Camelina microcarpa. As you can see here already, the seeds are quite different among these two species, and not only the seed size, but also the seed color. So we always like when the color are different. It means that many of the metabolites we are studying, they are also differentially accumulated. So what we did, so we use three different free Camelina sativa genotype and free Camelina microcarpa genotype that were sprayed with copper chloride or cultivated in control condition, and we harvest seeds at three different developmental stages. All these samples were subjected to untargeted metabolomic analysis and transcriptome. So the aim here is to see if we can induce the specialized metabolite in the seeds And what are the metabolic pathways that are, if yes, what are the metabolic pathways that are induced by the treatment? And if some of these metabolites can be somehow involved in pathogen resistance. And then, of course, we are really interested in the gene regulation part. So the idea is also to identify genes that are involved in the accumulation of these metabolites. And of course, you want to validate them through genetic approaches. So here you have the metabolic responses. Actually, there are the statistical analysis that take into account the impact of the genotype of the treatment or metabolite that are affected by both genotype and treatment at different developmental stage of seed development in the two species. So just one comment here, at 14 days after flowering, so the earlier, let's say, seed developmental stage, there is a stronger impact of the genotype on seed specialized metabolite diversity. So this is due to the fact that probably we started treatment just a few weeks before, so that the metabolome was not yet induced, let's say. And also what we can observe is that there is a much higher induction and diversity in microcarpa, non-domesticated species, compared to sativa. The situation is completely different at 35 days after flowering and dry seeds. In particular, at 35 days after flowering, there is a much higher number of metabolites that are influenced by the treatment, like that responds to copper chloride treatment, compared to the 14 days. And again, many of these metabolites were more different in microcarpa species. So what we did, this is just one of the questions was, are we able to elicit, to induce the specialized the metabolic responses in seeds by copper chloride? The answer, luckily for us, is yes. So here, each column is an average of 3 biological replicates of the 3 genotypes. So basically here you have 3 camelina sativa genotypes in control condition. Here you have the sativa genotype in copper chloride stress condition. Here you have the microcarpa metabolite in control condition and of the 3 genotypes, and here they are the same in copper chloride condition. So as you can see, so we have an induction here of many metabolites by copper in a camelina sativa, but more in camelina microcarpa. So that's nice, that's good. So one of the next step, of course, was to know what is inside here. So which metabolic classes are induced by copper chloride, either in sativa or in microcarpa. One more time, to make a long story short, so Nicola did a lot of work on the annotation of these metabolic classes. We identify one group of metabolites, one class, which are the cinnamic acids, which are massively induced by copper chloride at during seed development. Again, copper chloride was used to induce the specialized metabolome and to mimic a sort of pathogen stress. It's not exactly the same, of course. The idea is to validate these metabolites with a real pathogen, but it was much easier to do a spray treatment to induce the metabolome in this species. So basically what do we have is that copper chloride is induce a huge number of cinnamic acids in microcarpa, especially microcarpa, but also in sativa. And in microcarpa, we observe that 86%, which is really big, huge percentage of cinnamic acids are induced by copper chloride at this developmental stage. So very good. Then I went to Nicola. I asked, okay, which cinnamic acids are present here, since a very huge percentage is induced. So he did a very great job on the annotation, and he identified a large number of cinnamic acids, decorated cinnamic acid, that are induced by copper chloride. So here you have all the aglycons that we somehow identified. We have caffeic acids, coumaric acid, ferulic acid, synapic acid, and synapine. But of course, he didn't observe annotate only the aglycons, but he annotate also all the decorated specialized metabolites. So the next step for us from this side is, of course, to know more about the impact on decoration, mono D3-glycosylation, for example, of these metabolites on their activity against a pathogen. So next step. Of course, that's the question that we always we have in the team. So what are the genes controlling these metabolic pathways? So I received the transcriptomic and data like last week, I think, or 2 weeks ago. So it's a very, very quick analysis that I did. So this is just to show you that we observe a stronger induction of copper chloride of many genes by copper chloride at 14 days sorry, after flowering compared to 35, but which is not a surprise since the gene responses are normally earlier in the time course compared to the metabolites. And also, I did an analysis comparing just the metabolites that were more accumulated in microcarpa compared to sativa, and many of these metabolites were in the seeds were somehow related to specialized metabolite or specialized metabolic processes. In general, what we want to do now is to use all this data to trying to identify genes that are putatively involved in the accumulation of some of the cinnamic acids I showed you before. So here a few conclusions. So I show you that copper is able to induce the seed metabolome, which is already a good news because we were not sure at the beginning of the project. So we were very happy about that. That the metabolome of the non-domesticated species Camelina microcarpa is more strongly induced by copper chloride compared to sativa, and that the diversity of many of the cinnamic acids induced by copper chloride is huge, and also we think there is an import that we want to go at the decoration level, let's say, to study their bioactivity. So what is ongoing now? We want to study the impact of, we want to fractionate actually all the extracts that we did, and to enrich some fraction with the one that contains more cinnamic acids and derivatives, and study their impact on pathogens growth, in particular Alternaria brassicicula with our colleagues from Biogère. And then, of course, to identify some putative genes that are involved in cinnamic acids accumulation and induction, and there produce the mutants and study them. But there is still a lot of work to do, but I'm quite positive, let's say, about this project and about the results that we obtained so far. So here are some of the people that were involved in this work, but mainly from our team, Léa Barreda and Nicolas Clercoq, that did a very excellent job on the analysis and the studying of these metabolic pathways. So Léa is mainly involved in the first work on that was published in the Plant Journal on the diversity of seed specialized metabolites in camelina species, but he is also working in many other projects, especially on the impact on abiotic stress in Arabidopsis seeds. And she's also participating a lot to the second work, which is for the moment was mainly done by Nicolas, on the impact of copper chloride in camelina species. And also a big thanks, of course, to Stephanie, an engineer of the metabolic platform that did. She's involved in all my projects basically, and she did all the injection with the LCMS and analyzed a big part of the data. And of course, thanks also to the other colleague in our team, to Céline, engineer in CDD. Melissa, she's visiting PhD students in the team, and Mariana, that she just arrived as a visiting master student. Then, of course, all the other members of the team, Loïc, Delphine, Damaris, Céline, Sébastien, Martine, Bertrand, all the colleagues of other IGPB team that are collaborating with us, in particular the FIGER team, all the friends and colleagues from the IPS2 and the other colleagues from Italy or France that are collaborating with us. And thanks to you for your attention.
Thank you very much for your presentation. So now we will ask you some questions. Of course, you can ask if you prefer in French, if you're more comfortable. So maybe I will start.
Ouais.
We saw the impact of the environment on the plasticity of the seed, but do you observe this plasticity in other parts of the plant, or do you know if someone observes this?
Yeah, so we don't have the concerning the mimic biotic stress, let's go, it's copper chloride stress. We didn't do yet we didn't study yet the diversity of the analyze the specialized metabolome in camelina species. What we did with some colleagues from the IGEP Institute in Rennes, in particular Antoine Graveau, they did the same analysis in leaves and in roots. So they did the treatment in brassica plants, but in this case it was rapeseed in leaves and roots. And I also observed a huge induction of the metabolome by the copper chloride treatment. And what we did also with Lea, but there was more abiotic stress, but still it's part of the plasticity, of course. We, in Arabidopsis species, she did also targeted metabolomics on the leaves, and also there we observe a very strong impact on the environment in the vegetative tissues. So at the moment, to be honest, in the lab we are using the leaves induction of the metabolites to focus on the metabolites that are more specific in seeds. But of course, we can use the same data set to do other kind of analysis.
Okay, thank you. Eric, you can ask your question.
On vous entend pas.
Eric? Someone else have another question maybe for you?
Hello, yes.
Ah oui. Hello.
So maybe another question which is quite related to the previous one about the use of copper chloride, because I start a project about the use of essential oil to try to protect potato from mildew in order to replace maybe copper derivatives for the protection of potato. So generally, what is called bouillie bordelaise is a composition based on copper and copper oxides. What is known, what is applied, what are the potential perspective of using copper chloride, and more specifically in accordance with the problem of pollution due to accumulation of copper in certain areas, for example, in vineyard where copper derivatives are used regularly, and then we begin to see problems due to the accumulation of such copper into the environment.
So thanks for your question. So for the future, I don't think there is a future for copper chloride according to the legislation. So for this study, this one on camelina, the idea to use copper was really to elicit the specialized metabolome. So basically, what I want to see if what are the pathways that are induced by copper to avoid actually using copper at a certain moment. So here we will have also a genetic approach, so there will be all the production of mutants and etc. I have another project in collaboration with a colleague, Loïc Rajou from the FIGER team of IGPB, where we are comparing and the two projects actually. One is named Succeed, which is a project on aimed at substituting some pesticides or chemical compounds in agriculture for seed treatment. And there we are, and also another colleague from my team, Céline Rossi, she's involved, an engineer in this work. Here we are testing many different elicitors, not only copper chloride, to see if there are if there are other elicitors that have similar effects, so they induce similar induction. I'm thinking about Baba, for example, but there are many others. Some are commercial products, others are not commercial. And then, of course, this is more to search for substitute of copper chloride, that I think it will be banned in the next year. I guess that's a bit the tendency, or its usage will be reduced, at least for grapevine. And the second part is, of course, studying the fundamental processes that are behind the induction of these metabolites. But I think we didn't use copper chloride because we think there is a future. We use it because it was used since a very long time. So we are looking at the pathways that are regulated somehow by this product, but to search for substitute. Basically.
Thank you for your answer, for your presentation, and also thank you for the organizers.
Thanks. There are some questions in the chat. I can read them. Problem with microphone. Okay. A problem with microphone. My question concerns the genetic background of wild type, I guess, camelina. Are they selected homozygote? If yes, what about the metabolic diversity in wild population of camelina? So but basically, we don't use mutant for the moment, so they are all kind of wild type. What the difference is that for the camelina sativa I'm talking, they came from very different that's another question. They came from different breeding programs, and so the genetic diversity was relatively high, let's say, even if we are already always in domesticated species with lower genetic diversity compared to what we can observe in wild population. So basically what we observe in general, but we don't have the number to say that precisely, at least for the three genotypes of sativa and microcarpa, we observe a much higher metabolic diversity in the wild species, which is maybe not surprising. Then in general, I don't agree totally with the assumption that the wild species has more metabolites or higher diversity in terms of metabolome compared to the domesticated. I have the feeling, but that's a really personal idea, is that it depends to the class of the metabolite and on the species. There are some species where the domestication somehow increased the accumulation of some metabolites, and in other species they strongly decrease. Of course, for example, domestication decreases a lot the content of glucosinolates in many brassica species such as rapeseed, which is good for the fact that it's an anti-nutritional compound, but is not really good for the fact that it's also involved in biotic stress responses. So it will be nice to study more actually the metabolic diversity in these wild species. Next project, I guess. And Catherine, What about the intra-plant diversity? Intra-plant, you mean the same genotype or same species?
Yes, same genotype.
Okay.
From one flower or from one fruit to another, for example.
So sativa, there's no huge diversity in terms of within the same genotype. So the flowering is very it's constant among all the different plants of the same genotype. With Camelina microcarpa was a bit more heterogeneous, so we had to do a lot of tests before to find the condition to really standardize the growth of these plants. Something that I didn't say at the beginning is that Camelina sativa doesn't need at least the genotypes we are using do not need vernalization, and this is need for Camelina microcarpa. So we play a lot first with the vernalization time to some kind of synchronize the growth of the flowering, of microcarpa, but also to be honest, we have to kind of select some plants that have similar growth. So most of the plants has the very similar growth behavior, let's say, but it was a bit more heterogeneous. So, and also, okay, I tried to do the same with other Camelina species, and it was a nightmare. Like it's super heterogeneous, some were flowering, other no, and some were developing calyx, other not really. So I kind of stopped that part and I focused on more on the hexaploid, and was a bit easier. Yeah. So did you detect any phytoalexin in the Camelina species after copper chloride treatment? So in the seeds, we detect one actually, even if it's not super highly accumulated compared to what we observe in the leaves. What we observe a lot, of course, we observe a lot of these metabolites in our in the leaves and in the roots, also for the moment in the other brassica species in rapeseed that were induced by copper chloride. So we use this as a kind of confirmation that the treatment was somehow inducing these pathways. We also observe other metabolites that might be induced somehow to biotic stress, some related to jasmonate compounds or salicylic acid. So in the seeds they are present, but is much less accumulated compared to what you observe in the leaves. And also about the transcriptomic data, also there we observe some genes actually that were induced, that were linked somehow to phytoalexin, that were induced by copper chloride. So also that's, but again, is that we really received the data like 1 or 2 weeks ago, so it was too early to present them in detail.
Je vous remercie à tous pour vos questions et merci encore une fois pour cette super présentation. Je vais maintenant vous introduire le webinaire qui aura lieu donc le 2 novembre.
Ah, c'est Blandine.
Donc, vous voyez tous mon écran?
Oui.
Donc, pour le prochain webinaire, donc on accueillera donc une entreprise, donc l'entreprise Botanicert, qui nous présentera son expertise dans la certification des extraits végétaux. Donc, nous vous retrouvons le 2 novembre pour ce prochain webinaire. Donc, merci à tous encore une fois pour votre participation.